Well, good morning. It's March. I haven't streamed in a while, and I finally got an inspiration to kind of stream a little bit. Um, it goes on my thoughts and my thoughts only. Um, I'm not a scientist. I'm not a doctor. However, I do overthink things, but I also do believe that um, as a human being, we all need to make informed decisions and not just go with what everybody's telling us to do things or how anybody's telling us to do things. Doing our research is very, very important. So, um, that is really, really important. And how do we use informed decisions? How do we use our brain to make informed decisions? We use our critical analytical thinking. And everybody can do this. It's not something that, you can't do. It's just you train your brain to do certain things and read certain things. Um, I had really enjoyed one part of Mr. McCarn's stream the other night, and I asked him if I could clip it out and use it and put it up on my video because I think it would help a lot of people. But to have an informed decision, there's certain things going on that unless you know about this stuff and unless they look for things, you're not gonna find it. Um, and one of those things is, unfortunately, there's certain things going on that people are injecting into themselves and or haven't injected into them, that if you don't have an autopsy, it's just going to go down as a cardiac infraction, or what's better known as a heart attack, or clogged arteries. Um, this happens with the virus and in addition, and even more so with the virus because it contains the virus itself. However, there's something that, raised an alarm with me a couple years ago, or last year, and that was a paper on how the Chinese, and I don't care where it came from, okay, the paper, this is actually very relevant, found that the spike protein on SARS-CoV-2 without the virus RNA even is enough to do damage to our cells. And it's also not a respiratory disease as much as it's a cardiac disease. And if you listen to people like Kevin or Dr. Fleming is actually a retired cardiac physician. They can explain it a little bit more, but I'm not gonna do Kevin justice. So I'm gonna explain what I see, and if you have any questions, you can look all this stuff up for yourself. I just wanted to make a short video, as short as I can, explaining some of the things that he explained and why it's so important. Is that, like I said, most of the injectables, and when you're injected in things, aren't gonna really show up unless you have biopsies done that are evasive, or until somebody has a autopsy done. And it's not in everybody, okay? I think it, is in susceptible people that have the allergic kind of reaction to either the virus or the spike itself. So I am going to just go over here and read some certain key points on the paper that was done in April of 2021, which you know, with Kevin's got more things, you know, because he's looking for more followers, and he wants to get through to the population. Me, I'm just doing this for therapy reasons, and, you know, to get information out there to my family and friends. So I'm not going to do the paper justice because I'm not a doctor, but I do know some of the lingo, some of the language and stuff that's used. One thing I want you to really pay attention to is the inflammation that it's causing, and the term vasculitis and inflammation in general to our cells. I'm going to go to the paper now, and we are going to look at it together. Now, SARS-CoV-2 spike protein impairs endothelial function via downregulation of ACE2. Basically, the spike protein itself is impairing endothelial function, the downregulation of ACE2. That you'd have to, even JC, or yeah, Jonathan Cooey, you can find him too. And they, it goes on to say that SARS-CoV-2 severe acute respiratory syndrome coronavirus 2 infection relies on binding of the S spike protein to the ACE2 in the host cells. That's one way it infects. The other thing that happens is, it's really neat to learn when Kevin said that after it infects one cell, all it has to do is send out these filiopods, which is basically a you know, if you're ever seeing an amoeba with their pseudopods, you know, where they branch out. These filiopods are kind of the same thing, but only the filiopods come out and they poke holes in the neighboring cell, so the other, you know, the virus can get in that way too. But the binding ACE2 is what it does. Vascular, which means the heart and blood, endothelium can be infected by SARS-CoV-2, which triggers mitochondrial reactive oxygen species production and glycolytic shift. Okay. Now, vascular endothelium is basically heart tissue around different organism, you know, different organs and stuff, and the little cells can be infected. Now, mitochondria is an organelle. You think of our body as being systems and having organs in it, like the heart, the spleen. Well, when you get into a cell, a mitochondria is part of the cell organ, which basically is the powerhouse of the cell. And reactive oxygen species production. So anyways, ACE2 is a protective in the cardiovascular system, and SARS-CoV-1 S protein promotes lung injury by decreasing the level of ACE2 in the infected lungs. In the current study, we show that the S spike protein alone can damage vascular endothelial cells by downregulating ACE2 and consequently inhibiting mitochondrial function. They did a pseudovirus, basically, expressing the spike protein in hamsters, and they could see the lung damage was apparent in animals receiving the pseudospike, which is the fake spike protein. It's a spike protein without the virus RNA. It revealed by a thickening of the alveolar septa and increased infiltration of monocular cells. Now, that is basically what they're saying is thickening. Part of it is inflammation. Okay. Now, you go up, you read all this stuff, which is just a bunch of stuff that, like I said, if you need to know about this stuff, you can ask Kevin, Jonathan, or Dr. Fleming. In vitro means in test tubes, In vivo is in people. In vitro experiments using pulmonary arterial, EC is what? Endothelial cells, okay. Vascular endothelial cells. Pulmonary arterial vascular endothelial cells infected with pseudospike, which was rescued by treatment with N-acetyl L-cysteine, a reactant oxygen species inhibitor. Then they studied the impact of the spike protein on mitochondrial function. Confocal images of the vascular endothelial cells treated with spike 1 protein revealed increased mitochondrial fragmentation, indicating altered mitochondrial dynamics. All right, basically it's breaking down the cell and it's causing inflammation. This paper was done on April 30th, 2021. Now this is when we were rolling out our vaccine program in the United States. And when I say vaccine program, it's the mRNA vaccines that we're talking about, that have your body produce the spike, all or part of the spike protein. Okay, and that's what the mRNAs are doing. They can say it's not gonna, it's, you know, they made a pseudovirus. Okay, they can say they're harmless, but how do we really know? You know, as a population, we're trusting the scientists to do what they're doing. I mean, I have trust issues. That's why I go through these things and read and overthink. So this is me overthinking, but it's a good thing because I'm reading. I'm learning. Now, SARS-CoV-2 infection induces the endothelial cell, you know, cardiac endothelial cell inflammation, leading to endotheliitis, which means it's inflammation. Endothelial cells inflammation. Because spike protein decreased ACE2 level and impaired no bioavailability, we examined whether spike protein entry is indispensable for dysfunctional endothelium. The endothelium-dependent vasodilation induced by acetylcholine was impaired in pulmonary arteries isolated from pseudospike administered in hamsters. Although the use of non-infectious pseudovirus is a limitation to the study, our data reveals that the spike protein alone can damage endothelium, manifested by impaired mitochondrial function, which is in the cell. Okay, and this is a pseudospike protein that they made without the virus, and it's still impairing the mitochondrial function, which is an organelle, like an organ in our body, but only it's an organ in the cells, each and every cell that you have in your body. It's impairing that function to do its job. These findings need to be confirmed with the SARS-CoV-2 virus in future study. However, okay, a dysregulated renin angiotensin system due to ACE2 reduction may exacerbate endothelial dysfunction, leading to endotheliitis. Okay, it's just destroying cells basically. Collectively, okay, now this is where I had to ask Kevin. Collectively, our results suggest that the spike protein exerted endothelial cell damage overrides the decreased virus infectivity. This conclusion suggests that vaccinated generate antibody and or exogenous antibody against the spike protein not only protects the host from SARS-CoV-2 infectivity, but it also inhibits spike protein imposed endothelial injury. Now, this is a Chinese paper. You got to understand, they're not using the mRNA vaccines. They're using inactivated whole virus vaccine. The inactivated whole virus is actually in the vaccine itself. It's not forcing your body to make a spike protein. So that's what that comes from. I had to ask Kevin only because I wanted to be right. You know, I don't want to be talking out my ass with that. All right. So we see that the spike protein, they knew back in April 2021 that the spike alone, a pseudospike, actually harmed our cells and broke the cells down. Now, I'm going to show you this. This is very, very important because you're going to see stuff like this in the next video. This is the only tissue I want to show you. These are sections. The pseudospike is breaking down your cells. Okay? It's breaking down tissue. It's causing inflammation. You got lymphocytes and stuff in there. And to read this, you need Dr. McCarn or somebody else to read it. But basically, you know, I kind of know what I'm looking at, but I can't explain it good. But basically, you're seeing inflammation in a lot of, you're seeing a lot of the endothelial cells breaking down. Okay? Because you see, usually the stuff on the outer is on the outer circles, but look, it's congregated in the middle. That's going to be explained. See, there's nothing in the middle. It's all on the outside. This is the endothelial cell wall. Now, when you look at the pseudospike hamster tissue, you're seeing that the cell wall tissues are inside that. It's broken down, from what I understand. I might be wrong. Kevin can tell me I'm wrong. A couple people can tell me I'm wrong. But that's the paper I wanted to show you. Because, like I said, they were saying that further studies needed to be done. Okay? They need to be confirmed. And they need to be studied. And yet, they had this paper, and yet they still rolled out these mRNA vaccines, knowing full well that this happened, and not studying it more. Instead, they gave all the populace the mRNA vaccines. We do have vaccine injury happening. It's just being downplayed in the media. It's not everybody. Everybody's not going to get it, but a lot of people are. You're seeing people drop from sudden heart attacks, like people that you wouldn't think. My doctor is included in that. He had a sudden heart attack. He went out jogging, is what I was told. He came in and he collapsed on the floor. Heart attack. Now, because they're not doing the autopsies and they're saying, yep, it's a heart attack, or whatever, they're not even looking for whether or not the vaccine has caused any damage in these cells to cause that to happen. My friend, brother and son, okay, my friend is a girl, and her brother, but her mother's son, passed away recently. He didn't take any type of vaccine in him. Okay? This is why I say SARS can do this too. On a bigger basis. Alright? Having the infection, because he was not vaccinated. Alright? But this is basically what is going on in SARS too. It's breaking down the endothelial cells, and it's giving vasculitis. It's creating blood clots. It's really wreaking havoc in the body to susceptible people. Now, my friend was like me. He was sent home on, from what I understand, was told, Tylenol, cold and flu. Never had an anticoagulant or aspirin prescribed to him. It's a vascular disease. Doctors know this by now. My friend had high blood pressure and a heart problem. They didn't know the extent. But a red flag says, you have high blood pressure, I'm going to just give you some aspirin at the very least. And I'm not a doctor, but when my doctor prescribed, see, this is funny because it's good that I know this stuff, because when my doctor prescribed it and I have COPD, I was on that phone with my pulmonary doctor, and he said I was right. And I got the proper treatment early, and that's what they have to do. They got to do the proper treatment early for each case scenario. COPD is different. You call your breathing doctor. But, you know, if you have a heart problem, or even insofar as just, you know, high blood pressure even, you call your, you know, you try to get a visit with your cardiac doctor and see what he suggests instead. Because these people know full well it's what, 2022, April 2022. It's been a full year. They know that this is happening. They know. And yet nobody is getting angry that this was out back in April, and yet now we're seeing the effects of what this stuff is really doing. Like I said, I'm not doing Kevin or anybody, you know, any, what's the word for it? I'm not a doctor. I can't really think of the proper saying, but it really makes me angry. I talked to Dr. McCurn about this today. I go, you would think this would make people angry. And he's like, yeah, you would think, but nobody thinks that way. Right now, we're all focused on the Ukraine and stuff. And he's right. We're all moved on to bigger and better things. But this still is an issue. And I want to know why they didn't do more studies like that. What was this whole thing, just one big study for whoever was bringing these mRNAs out? I don't know. It should have been looked into more. Now, I'm going to shut my mouth, and the next video is Dr. McCarn's video. It's like 15 minutes. I'm almost done. And it's showing basically vaccine injury and what the spike protein had done to people that they autopsied. So, that's this next one, and then I'm going to leave it there. I'll come back and say goodbye. But I want you to really think about it. It really, really is kind of scary. Remember, they're thinking it's a BW.
The data is important because these people drop dead outside of a clinical situation. Okay? So, but they have been vaccinated. I would also say you can't discount the interactions potentially with latent SARS-CoV-2. There may be synergies that we're not aware of that emerge in some people.
Of the 15 cases that we examined 8 times by autopsy, 7 times in pathology, were always cases that were either shown as cause of death unclear or natural death. And in our subsequent examination by very reputed pathologists, as I said, we had a very likely context with the vaccination in 5 cases, likely in 7 cases, unclear, possible in 2 cases, and no connection to be determined to be seen in 1 case. Any pathology knows that beyond the 50th age, year of beyond the age of 50, deaths are usually multi-causal, and we don't say in these cases that people died of the vaccination. We only say that the vaccination had an influence on the death, on the occurrence of death, and in each individual case we have to perform give an expert opinion in order to make a clear judgment. We can't generalize here. I'll just show you some histology pictures. This is a normal thin slice through a liver tissue. We cut it into 1/1000th of a millimeter in order to be able to see them under the microphone. They are transparent, they're not colored. We color them so that we can see things, and what's important is the important parts are the dark spots here. We'll see that with the lymphocytes, and the cytoplasm is reddish, and sometimes you can see substructures even under the light microscope. Now the main findings, we have vessel lesions, the small vessels,
no, I've got everything sort of locked up on
coating
my PC
coating
for some
of the vessels, the endothelium, and I indicated in brackets the number of cases where we found this, and anthracite lumps, thrombocyte aggregates, microthrombo, and obliteration, so sort of blockages or obstructions of the smaller vessels. More seriously, of the major vessels, texture disruption of aorta and arteries with lymphocyte vasculitis and perovasculitis in 10 cases. And we can see how this examination is done. With the first cases that we performed, we thought, okay, this sort of thing happens. We have a patient by accident who has an aorta defect, but after seeing this regularly with these cases, we concluded that it must have to do something with the vaccination. We saw a dissection, a splitting up of the vessel in 6 cases, perforation in 2 cases, thrombosis without arteriosclerosis, perforations. We saw that in other studies, not in ours. And here are the images to corroborate this. On the left-hand side, you see the normal vessel. You have a coating with these flat spindle cells with this wallpaper-like cytoplasm. And on the right-hand side, you can see the same thing with the vaccinated person. You can see the spindle cells that have been detached from the wall. And then you can see these little dots. These are the lymphocytes that we will see again more frequently and that play a role here. This shows that it's not the consequence of degradation of the dead body, that this is actually something that happened in the patient while alive. What was particularly striking was the modification of the major vessels. Here we have a patient who died of a crack in the wall of the aorta, a rupture of the aorta, and usually the aorta has rather smooth, elastic layers. We can see it in the deeper layers, not at the upper layers where arteriosclerosis plays a role, but in the deeper layers we have infiltrates, lymphocyte infiltrates. You see the dark spots here, and what's even more impressive perivascularly with a vasovasorum, i.e. the vessels of the aorta, that's where we can see these dense inflammatory infiltrates. And here we can see these structural changes, these small ruptures, this dissection with lymphocyte infiltration again.
So he's describing all these darker purple regions here. And where the tissue has torn, the immune system is invading in to try to deal with any, cell damage, etc. Lots of stuff. So that's what you're looking for when you're doing these H&E types stainings.
Proof again that it happened in life, but not happened after death. We have lymphocyte infiltrates. And then
Now let's listen to this bit, because this bit is critical.
Weird findings, and I'll show you some other findings as well that I've never seen before, and that is very strange. We have this vessel with this strange material. I only took that picture yesterday, and I don't know what it's all about. This is an artery in the brain.
An artery in the brain. Now, it's fragmenting and would be clotted. So you're starving your brain of blood, oxygen, nutrients. Of course, you're going to get amyloid buildup.
And the interesting thing is that all the other arteries in the body that I looked at had no special finding. Only in the brain I have these strange deposits, and I'll speak about these foreign bodies later on again. So we were surprised to find these vessel modifications, and based on the fact that such vessel modifications can be observed with certain poisons, among others with so-called natural laterism, which is a poisoning in the context of certain chickpeas. That's why we performed some analyses in order to show the assumed toxin spike protein in those samples. And for those who are not familiar with that, we'll try to show this here schematically. As I said before, we have to dye the tissue, so we have a more complicated approach here. We have the spike protein on the surface, then we have an antibody for it, and then a further antibody, and we bind a dye to this antibody, and we can see this dye here. This is from a cell or a picture of positive cells that we were able to dye with the vaccination in a cell culture. And here we see our negative controls in the cell cultures. Here we have the proof of the spike protein S1 and in a larger amplification. Of course, we were criticized because it was claimed that if you say these were damages created by the toxic spike protein, it could be simply a so-called breakthrough, i.e. that the people didn't die of the vaccination or the consequences of the vaccine or the spike protein triggered by the vaccine. Instead, they had a breakthrough. They had a real corona infection. First of all, that's very unlikely because death by an actual infection, a COVID infection, without any preliminary symptoms, without you're developing a cough or something, you're simply collapsing at home on your staircase, is very unlikely. Nevertheless, we performed further tests to show the nucleocapside. Logically, the nucleocapside must be negative in the vaccination-induced lesions because the vaccination only provokes antibodies against the spike protein. So we have a cell culture that's clearly negative for that test. And now here's the findings proving the spike protein in the lesions. Here we have these positive reactions. That's exactly where we found the lesions, i.e. in the capillaries, in the interior wall layers of the small arteriola, and
again indicating that you're getting off-site expression. And everything about this spike protein just says it's cytopathic. And this data is very hard to argue with. I believe Joanna when she says that these people are some of the world's best pathologists. And even I can read this microscopy data. And yes, and I'm a fan of immunohistochemistry. I think it's one of the best tools that we have for tagging the presence or absence of particular proteins. And these people who have keeled over after vaccination are showing systemic damage. Everything that we've been warning about.
and then the aorta. This is the case with the ruptured aorta, and you can see here the leukoplasts that are clearly marked with the dye, accentuated by a vessel inside a vessel, and you have this clear accentuation here. And here are the results of our controls of the tissue. I showed it with our sample previously that we can only show the nucleocapsides with a genuine corona infection. Here's an image of the lung of a corona patient who passed with corona, and here we have a negative nucleocapsides test, and here we have a nose swab from a infected person with negative expression of the nucleocapside. Now this case is to serve an example of the modifications that the spike protein obviously provokes, leading to an incorrect, possibly an incorrect evaluation of the cause of death. With this patient, a heart infarction was diagnosed, and the microscopic view impressively corroborated this point of view. But fortunately, the coronary artery was cut histologically here, and we were actually able to show a thrombosis, which had led to an obstruction and an infarction. But what was strange was that there were lymphocyte infiltrates that are compatible with myocarditis, and what was also interesting that this not only had the thrombus and the arteriosclerotic modifications, but also these vessel texture disruptions that we'd already observed earlier, and these infiltrates that indicate periovasculitis that doesn't happen with a normal heart infarction, with a normal heart attack. So this apparent finding of natural death could be proven to be wrong with the histological examination. For those who are not so familiar with histology, a case of a patient who died of aorta dissection. You can see the surface of the vessel here. This is the vessel after being cut open. You can see that the blood used to flow around here, and you can see that there's a second and a third vessel wall. So the vessel has broken apart in the center. In the next image, we can see it even more clearly. We can see these two walls, and in the center, this bleeding that led to death. And again, we were able to show the spike protein in the cell walls. In the the cells of the vessel wall. On the left-hand side, we have the so-called fall aneurysm that can lead to bleeding, and on the right hand, what we just saw, the aneurysm dissecans. It's not the right term, really, aneurysm, because the vessel hasn't been widened. So you can see that the wall has been ruptured at one point. The center and outside layers have disintegrated, and the blood has penetrated here. And if the rupture happens externally, then the patient bleeds to death. And if you have this kind of vessel damage,
these are things that we have seen. And again, the question has to be asked, is how many is too much before we say, it was a bad idea?
Oh, gee, I was just talking without it on. Okay. Anyways, um, that cross section of the artery, I gotta go through this all over again. The cross section of the coronary artery, microscopically bothered me because that shows exactly what the first paper was talking about. It was talking about the breakdown of the cellular cell walls of the cardiovascular system, basically. And then the gross anatomy picture, the cross section, the last one, all that brown stuff was blood clotting. And that's what they're seeing in SARS patients too. Is everybody going to get it? Probably not. I think from watching a different video with a different doctor, they're finding out certain people are susceptible to it and certain people are not. However, because you can see this stuff happening, it makes more sense to use an aspirin or another more potent blood thinner in cardiac patients. Even though, like I said, I'm not a doctor, you'd have to ask Dr. Fleming this because he's a retired cardiologist and he's pretty smart. He's a really smart guy. And this information is just for making informed decisions and your critical and analytical thinking. I spoke with Dr. McCarn this morning and I said you'd think more people would be upset that they knew about this stuff way back in April of 2021, right after they were rolling out the vaccine, and yet you don't find a lot of information about it, studies that are being done, to confirm that the spike protein is indeed a safe thing, even if it's pseudospike. Now, they use the pseudospike. Now, I know that they say that it's, these vaccines are forcing your body to make all or part of the spike protein and that it's a harmless part. But, given the information that they just sent out there, with that last video, is it really harmless? Or does it depend on your body? I mean, vaccines, just like any other drug, isn't one size fits all. I'm not diabetic. If I take insulin, I might kill myself. I don't have, Tylenol doesn't work on anything for me, but Motrin does. But Tylenol works for other people. I mean, so it's like, it's a needle in a haystack for the doctors and stuff, and I understand that. However, we know a lot more, and for doctors to just prescribe Tylenol and send their patients on the way, I think, even we know too much about this to do that. So it kind of angers me that this is all now coming out, and I mean, they did numerous autopsies, that doctor that you just heard said. So anyways, I'm gonna call it quits. I had a faux pas in this video too. I keep on doing that stuff. So I hope you have a great day, a great week. Oh, tomorrow's St. Patty's Day. Oh, I'm mostly Irish from what my lineage is. I bought my daughter a nice Irish shirt for school, so we'll see. Happy St. Patrick's Day, and have a great day. I'm hoping you can get just information out of this. Like I said, one more tool in the toolbox. Have a great day.